Hello friends, today we are going to discuss about folic acid role in mental health. My name is Dr. Vijay Bhatia. I am one of the consultant psychiatrists. What is folic acid? The active form of folic acid in our blood and in our system is L-Methylfolate. L-Methylfolate is synthesized in the body from folate or dihydrofolate in the diet. That means when we eat food, it is normally in the form of folate or dihydrofolate, which is being converted into active form called L-methylfolate. It is an important regulator of a critical cofactor for monoamine neurotransmitter synthesis, namely tetrahydrobiopterin or BH4. BH4 is required by tryptophan hydroxylase to convert tryptophan into serotonin. This is another neurotransmitter which is, if deficient, can cause depression, anxiety, sleep problems, appetite problems. So it is important in the mental health. The other enzyme is tyrosine hydroxylase. This helps to make dopamine, and dopamine, as you know, is the reward neurotransmitter in the nucleus accumbens in the brain. And to convert or to make dopamine, we required BH4, and we learned that BH4 is generated from folic acid that we eat in our food, or folic acid is the synthesis form, crystallized form, whereas folate is the natural form. It is converted into dihydrofolate and then again into tetrahydrofolate and ultimately to L-methylfolate. Now, dopamine is again converted to norepinephrine, and this is noradrenaline, which you know is the fight and flight neurotransmitter required for a number of systems, raising your heart rate, increasing the pulse, supplying the blood to your brain to become more active so that you can fight the situation, stressful situation, or run away from it. It provides you that energy. Now, we mentioned that folic acid is the synthetic form, and what we eat in the food is normally dihydrofolate. But both of these need to be converted initially into tetrahydrofolate, and the enzyme required for that is dihydrofolate reductase. This enzyme is important because without the conversion of folic acid or dihydrofolate into tetrahydrofolate, the system will not proceed further because they are only one-way reactions. Now, these enzymes can be blocked by number of drugs. The most common is methotrexate. Methotrexate blocks this drug or enzyme that will not produce the dihydrofolate or conversion of dihydrofolate to tetrahydrofolate. As a result, the cell can die because methotrexate is the drug for cancer therapy. So these cells are fast-growing cancer cells, and if we block the availability of tetrahydrofolate, which is required for the number of mechanisms providing methyl group or one carbon moiety or molecule, and therefore it will stop. And the same thing happens in bacteria. Bacteria, they need the dihydrofolate to conversion of dihydrofolate to tetrahydrofolate, and if we block this by like trimethoprim, they will not be able to keep on reproducing, and therefore they act as antibiotics. And similarly, there are other drugs like pyrimethamine, which are used for malaria prevention. So this helps to understand that in the humans that these should be working properly and should not be blocked by other drugs to interfere with folic acid metabolism. Once the more active form of tetrahydrofolate is formed in the body, it is converted into methylene tetrahydrofolate, and the enzyme required for this is serine hydroxymethyl transferase, which is required for the conversion of tetrahydrofolate into methylene tetrahydrofolate. From methylene tetrahydrofolate, we convert the methylene tetrahydrofolate to L-methyl tetrahydrofolate. This is the form of folate or folic acid, which is in our blood, in our red blood cells, and it goes into the brain, into the neurons to help make other neurotransmitters like we discussed about dopamine or serotonin. Now, why I have put this as red? Because this is the enzyme methyl tetrahydrofolate reductase. In some families, this has been noted to have genetic mutation at 6, 7, 7 and 1296. What happens is there is a cytosine at the 677 allele of this gene, and this cytosine is replaced by the cytosine is replaced by thymine. And if there is a defect in this, which has been noted to be about 20% of the population suffering with bipolar illness or severe depression, meaning there will be defect in the conversion from methylene tetrahydrofolate to L-methylfolate. Now this is important because this is the one which is actually flows in the blood and is involved in the number of other reactions, which we will see in the next slides. Now, this one, after giving the methyl away, the enzyme methionine synthase will convert it back into tetrahydrofolate, and this cycle is repeated. And because of these important enzymes, if there is deficiency or a defect in these ones, they will cause problems in the mental health. Now we learned about the tetrahydrobiopterin, which is BH4, and we said this is a critical enzyme which is required by the hydroxylases, in this case tyrosine hydroxylase. This is a rate-limiting enzyme for dopamine, and norepinephrine synthesis, and even tryptophan hydroxylase for synthesis of serotonin 5-HT. Now, because L-methylfolate regulates BH4 production, it therefore plays an important indirect role in the manufacturing of these neurotransmitters, dopamine, norepinephrine, 5-hydroxytryptamine, or serotonin. Another important enzyme here is production of the glutamate. This is another neurotransmitter required in the brain, and this is the activating neurotransmitter which regulates the calcium flow into the neurons, activating neurotransmitter, and this is produced from histidine. And during this process, if there is lack of tetrahydrofolate, that will directly affect the production of glutamate neurotransmitter. This is another slide highlighting what we have already spoken earlier, that tetrahydrobiopterin plays important role. In the first instance, we mentioned about the aromatic amino acid hydroxylases, in this case tyrosine hydroxylase, tryptophan hydroxylase, phenylalanine hydroxylase. Now I've added this one new here is this is phenylalanine. This is one of the amino acid which is aromatic amino acid. That means it has a cycle or circle, and this phenylalanine is converted into tyrosine. Remember, when we were generating dopamine, it can be created from the tyrosine or it can be created from the phenylalanine. In the first instance, phenylalanine is converted into tyrosine, then tyrosine is converted into dopamine. And for both of these, we require the hydroxylases enzymes. This is also important in cuticle manufacture and melanin deficiency. If there is a mutant phenotype of the genes in the phenylalanine hydroxylase enzyme 1, then there will be problem in the cuticle. Cuticle is at the base of our nail, the skin, small part, which is normally can be dead tissue as well, and if it is also required for the melanin synthesis, and if there is a defect in this enzyme, there will be defect, melanin production will be low, and the skin will be less darker, and vitiligo or gray hair can develop. Now, tyrosine is converted into dopamine. We already discussed that. And tryptophan is converted into serotonin of hydroxytryptamine initially, and then into serotonin. And if there is problem with this enzyme, you will get the serotonin deficiency, and therefore can cause depression, anxiety, anger problems, emotional problems. BH4 is also required for the cell wall synthesis and fatty acids, and if there is problem with this one, there will be hypersensitivity and cuticle fragility. This is the skin at the base of the nail. It is also important in the synthesis of other enzymes, and it is circulated in a way so that it is kept on reproducing, and to reproduce this we require a continuous supply of vitamin of folic acid. I've mentioned here about remember when we were talking about the generation of the glutamate, which is a neurotransmitter, it is metabolized or made from histidine through a different cycle. The important bit I wanted to mention here is that in the process of this, we require tetrahydrofolate, which is folic acid. Low levels of L-methylfolate can lead to low synthesis of monoamines like dopamine, serotonin, noradrenaline. This can contribute to depression or to the resistance of some patients to treatment with the antidepressants, So if I give the antidepressant to patient with depression, they will not benefit from it because in the first instance, there is lack of enough monoamines. And for example, if I use selective serotonin reuptake inhibitors or selective uh, serotonin and noradrenaline reuptake inhibitors, what is the point of stopping the reuptake in the first instance when there is not enough supply of that? So therefore, these patients will not benefit from taking the SSRIs or SNRI. If there are no monoamines released, reuptake blockade is ineffective. However, repletion of monoamine synthetase by L-Methylfolate would theoretically make such patients responsive to antidepressant. So what we are saying here is that if you want to treat with antidepressants patients with depression, then if you give the SSRI or SNRI, if you add folic acid L-methylfolate to it, they may benefit more. Methylation of genes also silence them. Now what happens is genes, they can be silenced or they become active. They have like switches. What happens is L-methylfolate provides the methyl group for this silencing of the genes. So if L-methylfolate is low, potentially silencing of various genes could also be low. If the silencing of the gene for the enzyme COMT, which is catechol O-methyl transferase. Now this is the enzyme which breaks down monoamines, like dopamine is broken down by this enzyme, serotonin is broken down by this enzyme, and even noradrenaline and adrenaline are broken down by catechol O-methyl transferase. And if there is a low level of this, by silencing them, giving them L-methylfolate, and which will provide the methyl group to these genes, then the COMT will be low and more copies of this enzyme are made and enzyme activity goes up. So if folic acid is low, if we are not silencing this, the COMT will be high. The silencing of COMT will be low, therefore more copies of this enzyme are made and enzyme activity goes up, causing dopamine levels to go down, particularly in prefrontal cortex, potentially compromising information processing. This is the problem in patients with schizophrenia and depression, in dementia, or even autism, ADHD. In the prefrontal cortex, there is assumed that there is lack of poor supply of the dopamine. So hypothetically, if we silence this gene that produces a COMT, so what will happen? L-methylfolate could result in higher dopamine levels in prefrontal cortex, therefore helping with these mental disorders. What are the causes of low folic acid or low L-methylfolate, which is the active form in our blood? It could be dietary deficiency, people are not eating enough. In mental health patients, they don't have enough information about the importance of these vitamins, therefore they don't eat healthy food, meaning that this kind of vitamins are normally present in like green leafy vegetables, fruits, nuts. Mental health patients like schizophrenia, chronic depression, they don't eat healthy food or their appetite is suppressed. And if there are high levels of homocysteine, there will be a folate trap. And if there's B12 deficiency, that will lead to the folate trap. I have another slide to discuss about this, how the folate trap occurs when there is a B12 deficiency. What happens is folate, tetrahydrofolate is converted into methylfolate, and that is provided by the B12. and it goes in a cycle. If there is a folic acid deficiency, it can cause anemia. We know that in combination with B12, it can cause megaloblastic anemia. And what we have noticed is that there are genetic variants in enzymes that regulate L-methylfolate. And the first one is the dihydrofolate reductase. The other one is the methylene tetrahydrofolate reductase, MTHFR, which we mentioned earlier. What happens is at position 677, cytosine is replaced by thymine, and this will cause problems. And there's another site at 1298, adenine is replaced by cytosine, so that will cause the mutation, and therefore the enzyme will not function as it normally will. The other important enzyme is methionine synthase. When there is a adenine is replaced by guanine at the site 2756. Methionine synthase reductase is another enzyme. By the way, all these enzymes have been implicated in the genesis or causation of even schizophrenia. So if we inherit variants of these genes, what will happen? Leads to less availability of L-methylfolate, and this could potentially compromise monoamine levels by impacting their synthesis and metabolism. This will contribute to cause depression or symptoms of depression be linked to treatment resistance. Studies on these genes, variants of these genes, especially the methylene tetrahydrofolate reductase and catechol O-methyl transferase in schizophrenia, suggest that effects of some gene variants on the efficiency of information processing might be greater if variants in two or more particular genes are inherited together. So what are the risk genes and efficiency of information processing in schizophrenia? The single gene with the greatest risk may be the COMT, where there is valine replacement. Another one is methylene tetrahydrofolate reductase, which we already discussed in the previous slide. Risk may be even greater for individuals with multiple risk individuals who carry the neuroglin NRG1, ErbB4 and AKT risk genes. We will not go into these details because we are focusing on the folic acid here. Another important generation is S-adenosyl Methionine. L-Methylfolate, which is active form of the folic acid, is converted into methionine and finally into S-adenosyl Methionine, which is direct methyl donor for methylation reactions. If L-methylfolate is deficient for whatever reasons, there will be low levels of S-adenosyl Methionine, and it may be possible to administer methionine or SAMe, which is S-adenosyl Methionine, to such patients, and they can benefit from it. And they can also be benefited by giving L-methylfolate. However, if we keep on administering methionine or S-adenosyl Methionine, this will cause buildup of unwanted homocysteine that could theoretically interfere with epigenetic mechanisms and can eventually deplete precursors to the SAMe or S-adenosyl Methionine. Nevertheless, high doses of SAMe may be effective in augmenting antidepressants in patients with major depression. What is the recycle of the tetrahydrofolate by B12? Remember we said that methyl tetrahydrofolate is converted into tetrahydrofolate when methyl group is used by methyl in other reactions, and during that time, what will happen is the B12 is converted into methyl B12. This is cobalamin, this is methylcobalamin. Cobalamin is first converted to methylcobalamin by the N5-methyl tetrahydrofolate. And then what happens is the methylcobalamin is required for the conversion of homocysteine to methionine by enzyme methionine synthase. Um, so what happens is the homocysteine is being converted into methionine. So methyl B12 is used up here and give the vitamin B12, which is just cobalamin. And this methyl group during this process will be given back to the 5-methyl tetrahydrofolate, and it regenerates that. Now, this is a repeat. We have mentioned about BH4 is required in the conversion of the tyrosine to dopamine, and ascorbic acid is used in the conversion of dopamine to norepinephrine, and BH4 is required here as well. This is again we were talking about methyl or folate trap. What happens if there is a lack of B12? This is not being converted back into tetrahydrofolate, and there will be build up of tetrahydrofolate if the B12 is lacking, and therefore there will be a deficiency in the conversion of from homocysteine to methionine, and homocysteine can appear in the urine and cause other problems in the system. And this we already discussed, that it is required for the silencing of some of the genes. Folate receptor is another concept. Now what happens is the neuron or any cell membrane will not allow any molecule to enter into the cell until there is a receptor. What is a receptor is actually a gate. So if this is a cell, and it will not allow anything to go inside other than unless there is a receptor here, and this is folate receptor. And what happens is we have found that there are certain antibodies that they can attach here, and which will cause the folic acid or L-methylfolate will not enter the system, therefore the cell can die. And that's what is happening in some of the situations. Folate receptors bind folate. As we said, this is a receptor of folate, and it will bind with the folic acid of folate and mediates delivery of tetrahydrofolate to the interior of the cell. This will go inside normally. It is then converted into monoglutamates and then to polyglutamates and then stored in liver and other areas. However, if for this folate receptor antibodies develop, what will happen? This will block, the folic acid will not enter the cell, and therefore all the mechanisms of functions it does will not happen. And this has been noted in some of the tumors, epithelial-derived tumors, including the ovarian tumor or breast cancer, renal cancer. They can create these antibodies, and there are certain drugs being developed to help with these kind of therapies. Hence, antibodies to it are used in targeted therapies and diagnostic tests. And there is another drug that is being trialed for ovarian cancer, and it's already in phase 3. Antibodies to the folate receptor antibodies have been linked to the neurodevelopmental diseases. Particularly, there is one condition called cerebral folate deficiency. What happens in this one? The mothers gave birth. This is the mother, and this is the child. Mothers had normal serum folate, and even their spinal fluid or cerebrospinal fluid, they were normal, whereas the babies were born with antibodies in their brain, and they were found to have neurodevelopmental disorders, seizures, and they had learning difficulties, disorders, mental retardation. So folic acid is so important. This is again, I'm repeating here, cerebral folate deficiency in the brain causing all the damage, and there are certain other genes that will be affecting this and cause mental retardation in newborn children. This is the overall picture of folate being converted into tetrahydrofolate, being converted into methylene tetrahydrofolate, and methylene tetrahydrofolate reductase enzyme converting into 5-methyl tetrahydrofolate, which is then being used up in the conversion of homocysteine to methionine, making S-adenosyl Methionine, which are these are used in a number of other systems in the methylation processes in the body. So I'm not going to repeat here. It can, if there's a buildup of homocysteine, as we said, if there's a lack of B12 deficiency, this will not be converted into this and not produce enough of SAMe, and the buildup of this will cause the damage to the receptors in the brain, and like glutamate receptors, and there will be problems with that and can cause damage to the brain tissue. Folate deficiency due to drugs. This is important. There are a number of drugs we already mentioned. Trimethoprim is one. Pyrimethamine, that is antiprotozoal medication. Trimethoprim is antibiotic, and these can affect the metabolic process of the folate, leading to the deficiency and therefore leading to mental disorders. With other drugs which produce low serum and tissue concentration of folate, such as anticonvulsants, there are like sodium valproate, there are a number of other carbamazepine, and even antituberculous drug, especially isoniazid. Alcohol can damage. Oral contraceptives can affect even the absorption of these things. There are certain other foods like corn flour, maize. If you've been eating, the main staple diet is the maize, that will affect the absorption of folate in the first instance. Possible mechanisms include reduced absorption, prevention of released folate from tissue stores, altered plasma protein binding, or increased folate metabolism in the liver. Now, symptoms of children born to women who had low serum folate during their pregnancy. We have found they have given birth to babies which are low weight. They have developed neural tube defects, especially the spina bifida. Microcephaly is the small size of the brain, and they are born with a small size, and there will be developmental delays and seizures, fits, epilepsy, and they will have mental retardation. In summary, folic acid plays a vital role in the mental health, and because of the dopamine, serotonin, noradrenaline, adrenaline, they are important neurotransmitters. That means the one cell communicating with the other will be affected, and not enough information or good information is passed from one to the other. And if it happens in the children, they can cause a number of problems like spina bifida, microcephaly. And in especially pregnancy, it causes megaloblastic anemia. But make sure when you are treating megaloblastic anemia, we are giving B12 along with folic acid. Otherwise, there will be a folate trap, and there can be problems with B12 is essential for regeneration of methyl tetrahydrofolate. We already learned that, and we know that it is required for information processing in schizophrenia, autism, ADHD, dementia. Thank you for watching, and please press the like button if you like this, and also please subscribe the channel if you like it. Thank you for listening.